One of the male gamete fuses with the agar ovum to form the zygote. Another male gamete fuses with the two polar nuclei. What it is going to form? Triploid endosperm. So that's why here fusion taking place twice. One male gamete fuses with the female gamete to form the zygote. The zygote, from the zygote, embryo is going to get formed. Another male gamete fuses with the two polar nuclei. Then that fusion we are calling it as triple fusion. From that triploid endosperm, it is going to get developed. So here the fusion it is taking place twice. So we are calling it as double fertilization, and triple fusion in the case where one male gamete fuses with the two polar nuclei to form the so-called primary pen, what we will call primary endosperm nucleus, and again upon this the cell wall formation takes place, primary endosperm cell it is formed, and from this only endosperm it is going to get formed. So fusion of one male gamete with the egg cell it is called as syngamy, and fusion of the second male gamete with the polar nuclei which is present in the central cell to form primary endosperm nucleus. The syngamy and triple fusion occur in an embryo sac. The phenomenon it is known as double fertilization. Central cell with the pen it is called as PEC, primary endosperm cell, which develops into the endosperm. So the post-fertilization events includes the development of the endosperm, the development of embryo, and the maturation of ovule into seed, and maturation of ovary into fruit. Ovary develops into the fruit, ovule develops into the seed, and also the endosperm formation precedes the embryo development. The purpose is it provides assured nourishment for the embryo. Which is providing assured nourishment? The endosperm provides the nourishment to the developing embryo. So the development precedes, endosperm development precedes embryo development. There are three ways of embryo development. One is nuclear type, where primary endosperm nucleus divides mitotically without any cytokinesis, without any cell wall formation. That what we will call it as free nuclear we can call. Then the wall formation takes place, that condition what we will call it as cellular type. So if the endosperm it is completely utilized by embryo, the seed it is called as non-albuminous. If it is present, so such kind of seed it is called as albuminous seeds. So here that three types of endosperm example they have given. In case of coconut, if you see the kernel, the white kernel, it is nothing but the cellular endosperm. The water which we take, coconut water, that is nothing but the free nuclear endosperm. The cellular endosperm and also helophyll endosperm, in between this cellular and nuclear, the transverse wall and micropylar and cellulose chamber, etc. it is being observed in case of peas, etc. In case of castor, cellular endosperm, nuclear and free nuclear, and also cellular you can find in case of coconut. So this is about the so-called endosperm formation. So embryo development, then if you observe carefully, so from the egg, the embryo it is going to get formed. So it is going to divide into two, terminal cell and basal cell. So further it is going to develop into the so-called as embryo proper, and globular embryo, heart-shaped embryo, curled cotyledon, etc. All these, when it comes to the mature seed, in case of dicotyledon seed, you can find the embryonal axis. The embryonal axis, the region below this cotyledons, the the regions below this cotyledon we will call it as hypocotyle. The tip of this hypocotyle it develops into the root meristem, hypocotyle, and above that you can find the epicotyle, and it is going to terminate with the plumule. Hypocotyle, it is going to terminate with the radical. So these are the parts in case of dicot embryo. Then when it comes to monocot embryo, as there is only one cotyledon, it had been pushed to one side, the monocotyledon. That cotyledon we are naming it as scutellum. It is called as scutellum. Okay. So then it starts after certain embryo formation. The zygote divides mitotically to form proembryo, then globular heart-shaped embryo, horseshoe-shaped mature embryo with one or two cotyledons. It is going to get formed. Then coming to the next one. There are certain seeds are formed without fertilization. From the diploid egg cell develops into embryo without fertilization. If the cells of nucellus may develop into embryo and they are pushed into the embryo, especially in case of apomixis, what we will call in case of grasses and also asteraceae, you can find this apomixis. So, polyembryony in case of citrus fruits, if more than one egg may form into an embryo sac, so other cells like synergics or nucellus develop into embryo. Example, in case of orange, lemon, mango, etc. This polyembryony is observed. So, rest of the thing in the E content it had been given. Okay, hope so. I tried my level best to finish the topic in the given time. Thank you.